Good afternoon, everyone. So I'm going to talk about exploring post-transcriptional modifications during myrtle rust pathogen-plant interactions. So I'm just going to give a bit of an overview of the type of work that I do. So you may have heard there's all this genomic work being done on myrtle rust. Of the genome, there's 1 billion base pairs, and I'm looking at the RNA at the moment. Within all those 1 billion of letters in the genetic code, some of them are genes. Of these collection of words, we want to understand what they do. And this is kind of like when we want to look at RNA work, we want to have a reference transcriptome. And that's kind of like a dictionary. So of your genome, these parts of the genome have specific functions. And this is how we've traditionally done biology. And if we have a reference transcriptome like a dictionary here, we can perform RNA-seq experiments. And when we do an RNA-seq experiment, we have all these sequencing reads of RNA, and they're like the words. So in this example, it's a simple one. So the cat in the hat. We have some sequencing. We see the sequencing says cat, so it's a little piece of the page of a book. And we can refer that back to the dictionary, and we can tell what the cat is, what this gene is, and we can tell it's associated with MES. So we also see upregulation of this gene as well. However, the problem with RNA studies at the moment, no one's RNA-seq experiment looks like this. Everyone's RNA-seq experiments are actually really complicated. They have these very different stories to tell. And of course, nothing matches your reference transcriptome anymore. So Lord of the Rings, you've got Gandalf and Hobbiton. It's not in your reference transcriptome. Now we don't know what that means anymore. But we want to know what it means. So we look at it. It doesn't map to our transcriptome, and there's also other problems with this. So there's actually multiple isoforms. And of a gene, it transcribes into RNA. There's different isoforms in the sense like this, Gandalf the grey and Gandalf the white. Genes have produce different transcript versions, and they can have very different functions. Just like the wizard here can have very different powers. And within the cell, it can be located in different locations within the cell. And to actually perform RNA-seq experiments, we've had this problem where we use short-read sequencing, and these actually transcripts get broken up. So you can imagine if you break up Gandalf, you actually have a short read here of AND, and now AND maps to almost everything because the word AND in any story, it's very common. So how do I solve that? Well, what I've been working on is nanopore direct RNA long-read sequencing. And this is kind of a new technology when we can capture the RNA and we can sequence it through a nanopore directly. We capture the full transcript, so the full transcript, which has, and then all the different isoforms. And because we're measuring it directly, we can actually look at any changes within that transcript as such that transcripts can be methylated. This is kind of epitranscriptomics. So it's not only changes to bases or changes to the length of the transcript, but they may have chemical modifications. So the aim of the project that I've been working on, so first of all, we want to create a really good reference transcriptome for myrtle rust and other plant species that are infected by myrtle rust, identify the transcripts that are expressed and what's the response to myrtle rust infections, investigate if these isoforms are changing, so if they're changing from Gandalf the white to Gandalf the grey, and determine if these host transcripts contain modifications such as methylation. And in this experiment, I've got Syzygium jambos, so a Methylation plant, and infected it with myrtle rust, and also done a mock infection, so with no myrtle rust, and then I compare them. So what do I see? So when I do de novo transcriptome discoveries with this, with Syzygium jambos, a Methylation plant, I'm able to collect 33,000 genes. And within that, I actually see 124,000 different isoforms. So actually, there's a lot of isoforms for every gene. And I've also included Syzygium grande here, which is a publicly available one. And as you can see, they may have more genes, but they actually capture none of this isoform diversity. And I've done some trials with the myrtle rust as well. It's quite a big genome, and I can pick up, in just spores alone, I can pick up about 8,000 gene models. And again, there's a lot of different isoforms. So for every gene, there's actually multiple transcripts being transcribed. This is just an example of the complexity that I see. So the blue line is the gene, and then for each transcript we can have all these differences. So I can actually pick that up. So we could say, if we say the first one is Gandalf the grey, then I have also Gandalf the white, purple, black, orange, purple, and etc. I see many different forms of isoforms. When I look at what's changed, so once the plant has been infected with myrtle rust, I see all these transcripts being differentially expressed that weren't there before. One on the left is 3 days, so it's actually quite hard to pick up any changes at 3 days. But then on the right, I see at 6 days after a myrtle rust infection, I see all these red ones going up in expression and all these blue ones going down in expression. So ideally, the ones that are going up in expression are genes that are related to defense. And if we can capture these kind of genes and find out what kind of genes would be valuable for defense, maybe we can breed plants that have better defense to myrtle rust and to other pathogenic infections. And what I'm also seeing, so not only are the genes that are expressed changing, but you may have a gene that's always expressed, but it's actually switching from one isoform to the other. So going back to this Gandalf the grey and Gandalf the white, you may have one transcript that's expressed, but then all of a sudden myrtle rust comes along and infects you, and now you're actually switching to this other transcript, which we don't know the reason why it's switching or what that effect does, but it may be quite substantial because here in this Venn diagram I've got the black version. So more than 57% of the genes that I'm seeing in the plants that are infected, they have the potential to form multiple isoforms. And then at 3 days after an infection, I see 192 isoform switching events. So we kind of get this shift from one transcript form to another transcript form. And then at 6 days, I get 545 total. And these are really interesting because I'm seeing genes like cell wall modifying enzymes or chitinase degradation enzymes, which kind of suggest it actually is trying to fight back the fungus, but it's not quite getting there. This is an example of how I see plants responding with different isoforms. So at the beginning, I may have isoform X, and after infection, I kind of see one isoform going down, and then there's this trend of where another isoform starts to take over in the cell. And it's hard to know what this means, but as you can see, it's quite different. You have this, the green one has a much bigger 3' end, and this can actually be related to how what protein is made and where that protein is localized within the cell. And then I also see, even though I see things that are happening at 3 days after an infection, as the infection goes, I can also see changes again. So actually at 6 days, I see that isoform X actually goes down, and then there's this trend where isoform Z starts to take over. So in addition to looking at different isoforms, I've got this new really long-read RNA sequencing where I can look at methylation, so the methylation are chemical modifications to the RNA. And in plants alone, there's predicted to be 150 different RNA methylation events, RNA chemical modifications, I should say. And these can be related to stability, trafficking, the association with RNA binding proteins, and these can be really key to how the plant is actually fighting its infection, or if the fungus is actually doing something within the cell that can actually alter the plant RNA. And the reason this is actually really interesting because we have proteins that are actually writers. They'll write this onto RNA. There are proteins that are erasers that'll actually remove this methylation marks. And there's also proteins that are readers, so they can actually understand these methylation marks. And what I find really interesting with this is because you have RNA, and if you have proteins that actually can read the methylation there and actually cause a different effect or associate with that RNA and actually have a better defense response, that would actually be very beneficial for agricultural breeding. But also I don't really know if actually when the fungus infects, if it's actually expressing erasers and actually removing methylation marks that are on the on the plant RNA. And that could affect what the readers are actually doing. So when I perform these analyses and I look at the RNA methylation, I can detect about 400,000 sites, and that's of the two predominant methylation marks, M6A and M6C, 5MC. Most of these methylation marks are at the very beginning of the transcript, so what we would call the transcriptional start site or the 5' UTR, and that can be really influential on if that protein is actually if that transcript is translated into a protein and has a functional effect. And really interesting was, so about 80% of the genes that are in these Metaceae plants, they have the capacity to actually be methylated. When I look at the before and after infection with myrtle rust, I see very different methylation marks when it's not infected, and then after it's infected, I see very different methylation marks. And it is very interesting that actually plants that start to become heavily infected start to lose a lot of their methylation, which somewhat makes sense because actually the fungus doesn't really want the plant to be very happy in making its own proteins and living its own life. It actually wants the plant to provide nutrients and just be kind of static and just barely alive while it it takes over. So these graphs are really showing those kind of methylation marks before and after, and then for sites and for transcripts. So in conclusion, my direct RNA sequencing has revealed there is a complex transcriptome for Metaceae plants and for myrtle rust. There's widespread differential gene expression occurring, and plants are responding by using different isoforms. There's dynamic and reversible methylation occurring during myrtle rust infection. And our research goal is actually to increase our understanding of pathogen-plant interactions and determine if alternative splicing and RNA modification play significant roles in plant immunity. So I'd just like to thank the labs that I've been working with. There's the Justin Borovitz lab. So a lot of this work done today was actually the master student Xian Zhang, and also Scott Ferguson and Bhavika Kumar. Schlesinger lab, which is here today, especially Shenyan Lu, also helped us with the myrtle rust infections. And we do sequencing with Biomolecular Resource Facility at Australian National University. And some of this sequencing has been funded by Department of Agriculture, Forestry and Fisheries with the Forest and Wood Products Australia as a Science Innovation Award. So thank you for listening.
Thanks, Ash. That was really very interesting. Does anyone have a question? There's a microphone.
Sorry.
At day 6, defense-related genes were upregulated. What was downregulated? What was it shutting off?
Actually, the downregulated ones, I saw a lot of photosynthesis-related ones, which was quite interesting. So, I mean, the plant would still be photosynthesizing, but maybe not as much as what it normally would. So of these transcripts that are up and down regulated, I do want to associate them with gene ontology terms, which have been a bit tricky at the moment because we don't have a full annotation of all the plants and fungi we use. But ideally, I'd want to look at if it's energy related or defense related. But I had seen some that are actually would suggest they'd be defense related ones that are actually getting down regulated. So that would be good for the fungus if the plant defense genes actually were down-regulated, which could be actually related to the methylation. So if it's actually erasing methylation on those genes, it would cause that.
So I'm just going to ask as well, with the epigenetic imprinting of the methylation, it's not a question, it's just a comment, I suppose, but Do you see that perhaps looking at areas where they haven't yet got myrtle rust, looking at some kind of methylation patterns on plants that have pre-exposed, like naive, would inform? I suppose what I'm asking is the methylation pattern likely to be a static thing once it's changed? Is it irreversible once it's changed? I suppose not, but yeah. Once exposed to the pathogen, like so for WA where they don't have the pathogen yet, could you use this tool perhaps?
It is an interesting question. Probably don't know fully yet, but it does appear quite dynamic. But the interesting thing is, like, you have these read writers and erasers that actually can have very different affinities for different RNAs. So it can depend on whether, if the fungus is expressing erasers that are actually erasing a lot of things that actually, and then there's no way for the plant to recognize that that's occurred, and then for it to actually write methylation and then increase that transcript to be trafficked to the ribosome to be translated. Could be.